Apoptosis is a pathway of cell death in which cells activate enzymes that degrade the cell's own nuclear DNA and proteins. And fragments of the apoptotic cells then break off, as you can see here, and they would form apoptotic bodies. And this what gives it its name. Apoptosis means falling off. The plasma membrane of the apoptotic cell remains intact actually, so this membrane is altered to be degraded by phagocytes. These fragments actually call phagocytes to degrade them. The dead cells are cleared before contents have leaked out, so apoptotic cells don't really elicit an inflammatory reaction in the host, okay? This is opposite to necrosis. We have said in the first part of this lecture that it can be physiological or pathological, so apoptosis can be actually useful, and it's more useful than harmful actually. The first one is embryogenesis. What does this mean? During embryogenesis, you need cellular death to happen in certain areas. You can actually imagine that your fingers were not really fingers at some point when you were an embryo, and using apoptosis, these fingers got separated. Secondly is the involution of hormone-dependent tissues. Once you deprive a tissue from certain hormones, this tissue would shrink eventually. For example, the regression of the lactating breast after weaning. Lactating means breastfeeding breast. So whenever the baby suckles, this suckling stimulates the release of certain hormones that would eventually affect the breast and give it its volume. And whenever the baby stops suckling for a while, these cells are going to undergo apoptosis because there is no hormone stimulation. Third, cell loss in proliferating cell populations, like in your intestinal epithelia. You need to have a constant number of epithelia in there, and because of the activity and the function of these cells, you need to replace them every now and then, and they're going to be replaced by apoptosis. Fourth is the elimination of cells that have served their useful purpose. For example, when you have a lymphocyte at the end of an immune response, you no longer need this lymphocyte because it contains a microbe, so you need to eliminate it. And fifth is elimination of potentially harmful self-reactive lymphocytes. Self-reactive means that a lymphocyte that is willing to attack your own cells. So these lymphocytes are crazy, and it actually protects you when these are eliminated. Lastly, cell death induced by cytotoxic T lymphocytes, which is a defense mechanism that serves to kill virus-infected and neoplastic cells. Pathological causes are fewer. Whenever you have DNA damage, like due to radiation or cytotoxic anti-cancer drug, death would be better than a mutation that might lead to malignant transformation. Second is the accumulation of misfolded proteins, lead to endoplasmic reticulum stress, and apoptotic death would happen. Apoptosis can also be stimulated by a virus. And lastly, pathological atrophy in parenchymal organs after duct obstruction. This is very, very specific, okay? Let me explain this. Certain organs with ducts, they need these ducts to be patent, so they release all of their secretions. And whenever you have an obstruction, all of these parenchymal cells wouldn't really be able to produce anything, wouldn't really have anything to serve, so they're going to atrophy, and certain cells are going to undergo apoptosis, like in the pancreas, parotid gland, and even the kidney. Now for the mechanism of apoptosis. I know that this picture looks like something very, very complicated, so I tried to simplify it in this schedule, but still it looks very, very complicated. So let's talk about apoptosis as if it was a tale, okay? First, we have two pathways of apoptosis. The first is mitochondrial pathway or intrinsic pathway, and the second is death receptor pathway or the extrinsic pathway. Apoptosis is mainly intrinsic, okay? So let's start with mitochondrial pathway. Okay, so imagine that inside your cell there is a family that controls apoptosis. We call it BCL2 family. And it interacts with certain things that would eventually lead to apoptosis. So this family interacts with certain mitochondrial proteins. This certain mitochondrial protein would eventually initiate the work of caspase. Caspases lead to apoptosis. This family is divided into a pro-apoptotic side and anti-apoptotic side. Imagine that these are two brothers who have their own children with their own interactions, and whenever the pro-apoptotic side wins, apoptosis would happen, and vice versa. Okay, so pro-apoptotic family contains three brothers. The first one is the sensor. We call it BH3 protein. And then we have two twins. We call them backs and back. The anti-apoptotic part would also have its interactions, and we have actually two of them. The first is BCL itself, and the second is BCL-XL. Whenever there is a growth factor or something that stimulates the cell to survive, the anti-apoptotic part wins. BCL2 and BCL-XL would not affect the sensor, but would affect backs and back. And then, on the other hand, whenever you have something to induce apoptosis, like all of the causes that we talked about in the previous slide, the sensor is going to do its own work. It is going to stimulate backs and back and inhibit the anti-apoptotics. All of these interactions end up with the result of either stimulating backs and back or inhibiting them, because they do all the action. Whenever these are stimulated, they're going to work on the mitochondria. Inside the mitochondria, we have a protein that we call cytochrome C. Imagine that this protein is a dog trapped inside the mitochondria. So whenever backs and back are released, they're going to open certain channels inside the mitochondria to release cytochrome C. Cytochrome C, on the other hand, is going to stimulate the caspases to initiate the apoptotic cascade. And basically, caspase number 9 is the caspase that's going to start a cascade of nuclear fragmentation. And nuclear fragmentation is going to lead to apoptosis eventually. So this is basically the mitochondrial pathway, and this is how it is summarized in this schedule. Okay? The second pathway, which is death receptor pathways, is way easier. We have a TNF receptor family. This is a cell this is a very sad cell. It does not really want to live anymore. It wants to die. So it's going to express two receptors and is going to actually call a T cell. So this T cell is going to kill it. These receptors are only recognized by T cells. Okay, so you have your activated T cell, and it has a ligand to interact with these receptors and start a cascade of apoptosis by working on caspase number 8. And eventually nuclear fragmentation is going to happen. Now you can look at this figure and say, well, it's not really that hard. Okay, so remember when we talked about the morphology of the irreversibly injured cell and the morphology of necrotic cell, we also need to know the morphology of apoptotic cells. We can see that there are fragmented nuclei with condensed chromatin, pointed out by the arrows, with shrunken cell bodies, some with pieces falling off, and remember the word falling off means apoptosis. And the nuclear changes are chromatin condensation and then karyorrhexis. This is a schedule to compare necrosis and apoptosis. I think it's useful to read this. It's from Robin's Basic Pathology. Now we're basically done with the 3-part lecture. I know it's a long lecture. You can actually stop the video right here, but I just have 2 clinical examples that I want to talk about regarding cellular injury. These 2 examples are going to wrap a lot of things for you, starting from the first part of the lecture until this part. The first example is coronary occlusion, or what we call in more clinical terms, myocardial infarction. So you have a loss of blood supply, okay? So early on, the entire cell and its organelles are swollen, with increased concentration of water, sodium, and chloride, and decreased concentrations of potassium. If oxygen is restored, all of these disturbances are reversible, and in the case of myocardium, contractility returns. But if ischemia persists for a long time, irreversible injury and necrosis ensue. So you end up with severe swelling of mitochondria, extensive damage to plasma membranes, swelling of lysosomes, reactive oxygen species are going to accumulate in the cell, and massive influx of calcium may occur. So death is mainly by necrosis, but apoptosis also contributes. Remember, these 2 types of cellular death can actually coexist. The apoptotic pathway is activated by release of pro-apoptotic molecules from mitochondria, and the cell's components are progressively degraded, and there is widespread leakage of cellular enzymes into the extracellular space. Finally, the dead cells might become replaced by large masses composed of phospholipids in the form of myelin figures. Remember this? These are then either phagocytosed by leukocytes or degraded further into fatty acids that might become calcified. Another example is ischemia reperfusion injury, and I used the same picture because this happens whenever you have ischemia, but everything is still reversible. You did not reach necrosis. Imagine that this person went to the hospital immediately when he felt pain and he got treated. Now, this might happen whenever you have reperfusion. So, under certain circumstances, the restoration of blood flow to ischemic but viable tissues results paradoxically in the death of cells that are not otherwise irreversibly injured. Why? We have three reasons. The first one is that reoxygenation would increase reactive oxygen species from parenchymal and endothelial cells and from infiltrating leukocytes. So all of these cells are going to release all sorts of reactive oxygen species. And second, the inflammation induced by ischemic injury may increase with reperfusion. Whenever you have inflammation with good blood supply around it, which in this case is reperfusion, you're going to have more inflammation add on to that previous one. And third is the activation of the complement system, which is part of the immune system. Don't worry about this, but it actually contributes in the ischemia reperfusion injury. With this, we conclude our cellular death lecture, and we're done with cellular injury.